I think it's open. So today we're going to do sound or hearing. And again, it's one of the senses that is very much underappreciated. If you ask yourself if you had the choice between, missing sound, hearing missing, or missing vision, you'd probably say vision is, very, very, prefer not to lose vision. But imagine yourself without sound. And sound allows you to be connected to all your friends and all your relatives. And if you couldn't hear them, talking to them or anything, it'd be very difficult. And when children are hard of hearing, they often misdiagnose as having some sort of cognitive defect. And of course, sounds can save your life, especially if you're walking here on campus and texting at the same time and trying to cross some of the busy streets that the buses are taking people on. And then there's things like tinnitus, which is ringing of the ear. And it can drive you crazy if you have it. So what is sound? Well, sound is produced when something vibrates, like speakers in your stereo system and the vocal cords in my voice right now. And to produce sound in terms of my vocal cords, I've got to breathe out, and as I breathe out, the vocal cords vibrate, something like the speaker here. So if I vibrate the speaker, you can see I'm pushing these sound waves. And you notice that when the speakers moved, or the sounds moved through the air, you couldn't hear anything. You could only hear something when it strikes your eardrum. And sound travels relatively slowly compared to, let's say, light. So when you have a lightning bolt, you see it, and then only some time later do you hear it. Because again, sound travels a lot slower. So what we'll talk about is how that sound, which hits your eardrum, is converted to neuronal activity and finally conveyed to your cortex and allows you to hear. You can see that there's soft sounds and there's loud sounds. Loud sounds are produced by big vibrations of the speaker, and soft sounds are produced by little vibrations of the speaker, or little vibrations of my vocal cords. And then there's high frequencies and low frequencies. And again, you can see the high frequencies, the waves are closely spaced. Your speaker vibrates rapidly. And for the low frequency sounds, the speaker vibrates slowly. Now, you're able to hear anywhere from 20 to 20,000 normally. And as you age, this part here starts decreasing. So there are apps around, maybe I shouldn't tell you, but they operate at high frequencies, and students use them to signal to each other from their cell phones at high frequencies, which they can hear, but their prof can't. Now, the spoken word, that's covers only a small range of this frequency, 2,000 to 4,000. And our hearing is very tuned to those frequencies. Now, sound first passes through your eardrum, or your canal, and that's air-filled, unless you've gone swimming and it becomes water-filled, or unless wax gets blocks it. Now, one of the interesting things about your ear canal, that, again, this is FYI, you don't have to worry about it for the exam, is that the skin inside your ear canal grows slowly out, and then so it starts at the eardrum and moves over a period of a week out of the eardrum, out from near the eardrum, and that normally carries the wax out of your ear, but sometimes there's a big buildup and it blocks this canal. So that's the first part. Normally that's air. And then there's the middle ear. That's between here, this drum here, and this smaller drum here. This is your eardrum, and what you see or what the doctor sees when he looks in your ear. And then connected to between these two drums is a set of bones, and they're called the ossicles. And they transmit vibrations from this big one to this little one. And then over here, we've got the middle ear, which is fluid-filled. Okay, and inside this eardrum, this inner ear, you have this cochlea, which is, cochlea means snail, and you can see it looks like a snail shape thing. And inside that is a little ribbon that goes round and round, called the basilar membrane. And we'll see in a moment that that contains then the neurons that get activated by the vibrations. This desk is sticky, so my mouse is sticking on the desk. So you can see here the air or the sound coming down the ear canal, pushing on the eardrum, and then pushing on this other window, little drum here, which is called the oval window. And again, the ossicles acting as a lever from one to the other. And over here, this cochlea, I've unrolled it to look so it looks more like a can than the cochlea really looks like. But it's easier to draw things within it. Now, air is like a light molecule. You can think of it as a light molecule. And water, you can think of it as a heavy molecule. And when a light molecule, like a ping pong ball, strikes a heavy object, like a billiard ball, it'll bounce off. Okay? So very little motion will be produced inside this fluid container, and so you won't hear those vibrations. So what the ossicles do is they act like a lever system, so that this little light molecule can lift this big heavy molecule and cause changes inside this cochlea. Now, attached to the ossicles is a little muscle that the brain can contract. And when it contracts that, it reduces the movement of the ossicles and protects your ear from damage. And the muscles here on the ossicles contract for very loud sounds, like when you enter a bar that's playing loud music, or you put your earphones on too high. Or, surprisingly, they contract every time you speak. Okay? Your own voice is really a loud, loud, loud sound to your own eardrum. So it responds sort of protectively to everything that you say. Now, what happens here? Okay, in this cochlea, we have something called the basilar membrane, which is this ribbon lies from one end to the other end. You can imagine that this is something like a long rope that I have, that I'm holding one end on this side of the room, and the other end is fastened to the wall at the back of the room. And I flick it, okay? And when I flick it, this vibration will start here and end up over there on this rope. Well, we want this vibration here to start occurring at this point here when we press down. Now, this is all fluid filled, so it needs something to escape, and that's produced by this round window, which allows pressure here to deform the basilar membrane here, and pushes on the round window here. So like a release valve. Now, on the basilar membrane, you've got these really amazing things called hair cells. And every time the vibration passes down the basilar membrane, these hair cells shake, and they get bent. And when they bend, little strings, tiny little strings, open up little flaps at the top of each hair. And when they open, potassium enters the cell, and it causes the voltage to rise, and that increase in voltage causes this cell to produce more transmitter and produces action potential in this 8th nerve here. Now, you can imagine that this is a tiny microscopic cell, and on it you have even smaller microscopic hairs, and attached to each hair you have a string with a tiny, even more microscopic flap. So you can imagine how delicate this instrument is, and why these hair cells can be damaged by loud sounds. Now, the reason that this flap is built is because you can open and close it very rapidly depending on how the vibration is coming, and so you can quickly depolarize it and repolarize it depending what is required. That's much faster than a channel opening up chemically. Now, when this basilar membrane bends, this flick of this cord occurs, and it starts traveling to the other end. Okay, but the bulge or the deflection in this cord is interesting because it doesn't stay the same size. It increases and then decreases. Now, the increase and decrease is dependent on what the frequency of the sound is, okay? So you can see that over here, it's loud, high frequency sounds that push this end more, and low frequency sounds push this end more. So it's like a piano, the strings of a piano. These are the high frequency strings. These are the long, low frequency strings. So here the strings are wide and floppy, and here the strings are narrow and stiff. And therefore the vibrations produce more high frequency displacement, displace the hair cells located here more, and the low frequency sounds displace the hair cells that are located here more. So those are 3 notes that were played, and those 3 notes you hear because hair cells over here, then hair cells over here, and then hair cells over here get activated in the right sequence. And so it's not that the hair cells are sending a certain frequency to your cortex that you hear a high pitch or a low pitch sound. It is that a particular hair cell is being activated. Okay, so the ones, if ones here are being activated, then you hear high frequency sounds. If one over here are activated, then you hear a low frequency sound. And you've got something like 16,000 of these hair cells strung out along this cord. And so they act like a labeled line. Remember, you sensed that something was a certain feel of touch depending on what afferent was being activated. Well, now it's not what type of quality of touch, but what frequency it is. Now you can notice here that hair cells over here got activated each time, but to a different amount. So when it was a loud sound, you had a big vibration here, and was a low soft sound, you had a little vibration here. So loudness is produced by how much frequency is being generated, how much action potentials per second the hair cells here are producing. So frequency is by which hair cell is firing, the loudness of a sound by how many action potentials per second that neuron is generating. And so when you hear a more complex sound like that, well, that's because there are many hair cells firing at different times. And so this basilar membrane is acting like a synthesizer. It's producing this much of this amplitude of sound at these frequencies and this amplitude of sound at these frequencies, generating these complex sounds. So it's decoding all the sounds that are being sent through your eardrum. Now, your ear is a very delicate structure, and it can get damaged by for a variety of reasons. Very loud sounds like a gunshot or an explosion can tear the eardrum. It can break these ossicles, or it can damage your basilar membrane. Less loud sounds can break the hairs on your basilar membrane. If you go to an audiologist, they test what frequencies you've lost. So they do play each frequency one at a time and see how loud a sound, what the lowest sound you could hear at those frequencies, and can determine what frequencies you're missing. And they can tell you if you've been someone in a rock band, because that'll damage the low frequency sounds, because those are usually played the loudest. And they will break the filaments on your hairs. And the same thing will happen if, of course, you play your earphones too loud. The other thing that happens often is that you have an infection, and fluid will fill this middle ear. And the doctor, when he looks into your ear, will see a bulge of the eardrum. And that'll impede your hearing, somewhat similar to when if you go swimming and water fills this outer ear canal. Okay. So the same thing. And also infections can affect the hairs in your hair cells. Now, interestingly, these flaps that open up and close allow molecules, not just potassium in, but molecules of different sizes, or at least of relatively small size. And early antibiotics that were used to treat ear infections used to enter this channel here and kill the hair cell. So people went deaf from some of the early antibiotics, but now they've developed types that don't affect the ear neurons. And of course, people like me, you can lose your hearing simply because it wears out. Your blood supply affects your hair cells, and once they're lost, they can't be replaced. Unlike your taste and your smell afferents. Okay. Besides volume and besides frequency, there's another quality to sound, and that is the direction. We've got two ears, and that allows us to hear location, hear what location sounds are coming from. So like the two eyes that you have allow you to see in stereo, the two ears do the same thing. It's working. So you can hear how the sound is moving from one side of the room to the other. And in this case, what's producing that change is the loudness of the sound from one side to the other side. If the sound is coming from let's say the right side, then this ear hears it full force, but this ear lies in the shadow. Now that works best for high frequency sounds. Low frequency sounds can wrap around the head and activate both ears very similarly. The other thing that the brain uses is the timing difference between the two sounds, two ears. So here we see sounds hitting this ear first and this ear second. And so the brain can tell the sound is coming from this direction, a little to the left, or my left, your right probably. Now the difference here is huge. It's not what really happens in real life. Sound travels very fast. So this sound wave here is traveling at like 500 miles per hour rather than this slow speed that you see here. And so it passes from one ear to the other in something like 10/1,000,000th of a second. So it's remarkable that the ear can detect such a small difference. And the way it can detect that is largely because of those flaps that are opening and closing. And again, this difference in sound is best for low frequency sound. So it uses one cue, the loudness, for high frequency sounds, another cue, the difference in timing, for low frequency sounds. Now, the other contributor to location is the shape of your earlobe. Now, there's a reason you all have these funny things on your heads called earlobes, because first of all, you hear things better when they come from the front than from behind you. But you also hear things different when they're coming from above or below you. Okay, that's in part because within your earlobe you have different shapes and contours, and they produce sort of bouncing of the sounds that occur in your earlobe, and so you have little echoes, and from those little echoes the brain can determine whether or not the sound is coming from above or below you. And finally, what you can do is you can turn your head and you can localize sound to when it's coming equally to the two ears. So when you turn your head towards the sound, you'll again notice that it's coming directly in front of you by the fact that the two ears are activated equally. Now, other visions also help you localize sound. Vision is a good quality that causes you to mislocate where sound is coming from. And ventriloquists will use that to misdirect you to where you hear the little toy or something that the ventriloquist is opening the mouth of. That the same effect works in movies. When you see lips move, you localize the sound to that person on the screen. Now, the first place that the brain can compare the two ears is the structure here called the superior Oliveri nucleus. So this is where sound from one side first gets together with sound from the other side. Now, this diagram here, there's a mirror image of this diagram on the other side. So there's one superior Oliveri nucleus here, and there's another identical one over here. Within each one of these, within each Oliveri nucleus, you have two parts. A lateral part that codes the intensity differences between the two ears, so it compares that quality, and a medial part that's looking for the timing differences between two ears. Then that information is sent first to the inferior colliculus, and that connects to the superior colliculus, and that allows you to turn to whenever you hear a sound. Okay, so if a sound, you hear the door slam, you'll turn your head towards that sound. We'll cause an orienting response. And then the signal will also go to the thalamus, in this case the medial geniculate, and then from there to the primary auditory cortex, where you'll be conscious of hearing that sound. Now, a question. Okay, so let's suppose you hear this type of sound. So you hear, this is 5 milliseconds, so each of these about a millisecond apart. They hear this, something going louder, 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 more action potentials. Shouldn't have said louder. That's giving things away. So more action potentials. So you're measuring a neuron, and this is what it's doing. So is it 5 similar sounds played every 1 millisecond? 5 sounds of an increasing frequency? 5 sounds of an increasing intensity? Or 1 sound of 1000 hertz played once? Or 5 sounds played and shifting from the left to the right? So I'll go through. Is it this one? Hands up. This one? Hands up. This one, hands up. Oh, good. This one, hands up. And this one? I'll go yes, because I know what the answer is. Yeah, no, it's both of them. So it could be this, because as it goes from one ear to the other ear, it might be getting louder. It might be, in this case, it was facing. This wasn't the ear that was facing it, and then over here it was the ear that was facing it. But yes, this is the same. So the same hair cell is firing, and if it's the same hair cell, then it's the same frequency. All it can do is change how many action potentials it's producing, and that's produced because the sound gets louder, either because the sound itself gets louder, or it turns from one ear to the other. Okay, so what happens when it enters the primary auditory cortex? Well, within the primary auditory cortex, you've got columns. And within each column, you've got different frequencies encoded. So it's as if you have your basilar membrane mapped onto your auditory cortex. And you begin at one end at low frequency sounds and at the other end at high frequency sounds. So you have this tonotopic representation. Now, all sounds will activate this primary auditory cortex. Around it is another area called A2, which is activated by sounds that are word-like in quality. And for example, these sounds are called phonemes. They're parts of words. Ba, ga, r sound, l sound. These are all used to compose words. Now, this A1 is called the core area. This A2 is also called the belt around it. And together, these two areas actually contain within it a dozen or so maps within it, each one doing this mirroring that we saw both in the visual cortex and again in the sense of touch. So it's not just 1, 2, or 3, but it's a dozen mirrorings in there. Lots of stuff going on. Finally, from there, sound is sent to a structure called Wernicke's area, which is used for word comprehension. So it's where you start understanding what the meaning of the word is. Now, newborns, and it doesn't matter where you're born, whether you're born here in North America or on the opposite side of the world, you hear the same set of phonemes, the same ba, ga, r, l sound, everything. And then Patricia Kuhl, I've got a link to her talk that you might like listening to. She found that after the age of 6 months or so, the auditory system starts to filter these word-like sounds and bunching them up. So try saying, all of you, try saying ba, ba, ba. Now say pa, pa, pa. Okay, notice that there's a very slight difference in those two sounds. Ba and then pa. The difference in how you explode it, you know, from your mouth. And that difference is sort of is heard because this within this system, you start saying, okay, that sounded more like a ba, and this sounded more like a pa. They act like magnets, attracting familiar sounds. And there's a clear boundary between. You either hear a ba or a pa, nothing in between. Now, how do they know that everyone in the world, or every baby in the world, okay? So what you saw here was some ba sounds started to play, and that baby got woken up to this pa sound, and it started sucking on this pacifier, which then was recorded by this monitor. And then gradually the baby got bored by this sound and fell asleep. And then when this sound changed from pa to another different sound like ba, it woke up again. It was stimulated. Again started using this pacifier, and again that was recorded. So Patricia went around the world and measured this activity coming from babies, both babies before they hit 6 months and babies after 6 months, and found that there was this gradual transition. So if they developed an ability to hear sounds that existed only in their own environment. So if you were raised in an English environment, you would hear an R or an L again. Try saying L, R. You can see you have to put your tongue in a slightly different place for the two of them. And if you slide your tongue back and forth, it becomes either an R or an L. In one case, the tongue is in front, and the other case, the tongue is further back. And there's nothing in between. Now, if you're born in an environment, for example, a Japanese environment, you don't hear on these two different sounds. Only one of these is produced. And so as a consequence, both these sounds are heard as one. And you lose the ability to distinguish between these two. Similarly, there are phonemes in the Japanese environment that someone who's grown up here in North America, it becomes blind, auditorily blind to. Okay, so what sequence of areas get activated when, for example, you read something out loud? Okay, I'm looking at the screen and I'm reading the things out loud. Well, first of all, you activate your primary visual cortex, and that, of course, get activated by lines, and the lines that I see on the screen. And that goes to these other areas. For example, one of them is this visual word form area that we talked about in, I guess, the third lecture. And that is where the left and right side of words are put together. If you look at a word, you're usually looking at about the middle of the word. Okay, so that means one half of it is projecting to one cortex, the other half of it is projecting to the other cortex. The two halves have to be put together, and they're put together in this visual word form area. And that's located in the posterior end of this fusiform face area, but on the left side of the cortex. Okay? So it looks like this fusiform face area, the area that normally gets activated for faces, becomes taken over by this area that we use to read. Now, reading just developed recently in human history. I mean, it did develop 2,000 years ago, but very few people read 2,000 years ago. Very few people read 200 years ago. It's something that we are not genetically pre-programmed for. There's no gene for reading words. So it's something all of us develop by practice. And what we do is the brain has taken over this little part of the brain, little area called the fusiform face area, and said, okay, I can use this area that was good for faces for words. And words and faces are very similar because you recognize a face instantly. You can tell if it's this person or that other person instantly by taking that whole face in. Similarly, you understand a word the same way. When you see the word, it instantly gives you a meaning to it. Okay, that combination of letters is like this combination of features on a face, and you have to train it by repetitive, activation from childhood as you grow up. And if, of course, something is wrong with this visual word form area, then you develop dyslexia, like you would get prosopagnosia if you had fusiform area problem. These two things often go together, so people that have trouble recognizing faces usually also have trouble with reading. Now from there, you go to the PTO association area. That's relatively poorly defined, but the reason it's located here is because you can put together things the words that you can see, or you can hear, or you can feel by touch, as you do in Braille. Okay? And you can recognize that something's an apple by its feel, or even by its taste. From there, we get to this Wernicke's area that sounds coming through the auditory system land in. So here we're getting visual information. Here we're getting auditory information. And finally, oh yes, I should also mention that the biological motion. Remember the superior temporal sulcus, okay? That's where we had visual information that related to biological motion. Well, one type of biological motion is lips moving. And we usually recognize what people are saying by watching their lips carefully. And people that are deaf learn to recognize what people are saying just by their lips. I won't play this thing, but if you do do this at home, you'll get an amazing video of this person saying ga, 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 and you're hearing it as pa, pa, pa because of his lips. It just changes because you see his lips doing one thing, but that's not what he's, if you close your eyes, you'll find that's not what he's saying. Okay, from there, information flows to Broca's area. Now, this is the central sulcus, so this is all the frontal lobe. Okay, so the frontal lobe, you have working memory, and working memory is important in putting the order to words. So your ability to say things, starting with, in English, the subject first, then the verb, then the object, then the right order. So you have things that dealing with grammar are dealt with over here. And then finally we go to the motor cortex, which is located here just in front of the sensory cortex, somatosensory cortex, and here down near where the auditory system, near the sylvian sulcus, is where the face muscles are, and the muscles that contract your lungs are. And they then produce the sounds that you hear. Okay, so if you have damage of these areas, you've got different problems. If you have damage in Wernicke's area, you can't understand language. Okay, but because Broca's area is still intact, you can still say things, but often they're nonsense because there's no meaning behind it. If, on the other hand, you've got a lesion in Broca's area, you won't be able to say things in the correct order, so your sentences will all be grammatically incorrect, but you can hear what you're saying, and as a consequence, you can get very frustrated because what you hear is not what you want to say because you've got this damage. So the other thing is the sign language follows the same pathways. So people that are deaf use sign language to communicate. And sign language is composed of gestures with the hand. If different gestures mean different words. And again, if you have damage to Broca's area, you'll lose the expressiveness of the hand gestures. Whereas if you have a damage in Wernicke's area, and you're with deafness, you lose the comprehension of the gestures. So these areas are not just limited to sound, but to things like moving your hands and the vision of moving your hands. Now there's also an auditory what and where. The what pathway heads in this forward direction and then to Broca's area, and the where pathway heads back into this area here, our understanding area, and then flows forward. So if you have the auditory what stream tells you the identity of a sound. So if it's who is speaking, or is it the sound of a lion? Okay, so that's why it's called the what pathway. What is it? Whereas over here, the where pathway is used to tell, first of all, the location of a sound, but also its temporal properties, and as a consequence, what the word means, or if the sound is a word, what its meaning is. And the children, start babbling, even at the age of 6 months, and this babbling, involving productions of sound, helps them develop language. I think that's it. Thank you very much.